I'd like to spend a little time talking about the microbes and just give you some of the new idea about what people have been dealing with the HGT in bacteria. And in bacteria, we know that it's very easy for them to have conjugation or transformation, and the genome are usually network-like. So this is the hypothetical relationship among the different microbial genomes. And you can see, well, they do have a lot of horizontal transfer in all the cases. And the bacteria, even within the same species or among the different species, they very common to form this kind of channel, the nano channel, like the pili. And then it could be very easy to transfer the gene material from one to another. And there are several mechanisms, of course, and we just mentioned that there's transformation, which means that the material will directly taken up from the environment, and the naked DNA was integrated into the genome. That's called the transformation. And the conjugation is usually to replicate the plasmid and through the channel of the pili, and then copy that. And also it could be carried out by the vector, like the case of the phage. And that's called the transduction. That's to, there's an eruption for the cell, the whole cell to the donor, and then to have the DNA transfer to the recipient. And there's some cases right now recognized that there's something called the GTA, the gene transfer agent. They will transfer the DNA without killing out the donor cell. And there's some cases supporting this kind of idea. So there's some very interesting discoveries in recent years that they want to visualize and quantify how many gene transfer actually happen. We all know this kind of mechanism, but how often do they happen? Can we visualize that? And so they use this ICE as the case, and this is the ICE is called from the integrative and conjugative element. And this element will excite itself and to become a plasmid-like thing. And it can through this channel and to replicate themselves and to have two different copies on the donor and the recipient cell. So if we can check a certain piece of DNA from the donor, and then we can probably check the thing to the recipient, and they'll have two copies right here. So we can probably visualize those kind of events. And this is the system by that. It was published in recent year. And so basically, the donor cell has this red fluorescent protein. So anyway, so it's the color will be red, and the recipient is green. And they try to use a reporter gene, the GFP, and they use this spatial construct to from the donor and to the recipient. In that, it will this particular case, it will bind to the leg guy gene. So, which means that if there's the IC elements transfer from donor to recipient, and then the protein will bind to the recipient genome. And because it binds to a particular region, the color will be lighter compared to the other part. So as long as you can find a very green spot on here, then you will see, you will know the gene has been transferred and integrated to the recipient genome. So this is the basic idea. So they can use this real time to monitor this event, and they can monitor this lightening of this thing. You can see from one cell to another, and basically within 30 minutes you can transfer the gene, and then integrate to another genome in 30 minutes. So it's very fast actually. And in their study, there's more than 80% of the recipient will turn into a donor very quick and transfer that. So it means that the conjugation could be very fast. We will then expect that the bacterial genome will be highly dynamic. And in recent years, there was some group developed something called the phylogenomic network. And the basic idea is to compare the different genomes and to compare the particular part of the genome to see if they are similar to the other taxa. And then they try to use this special network and to reconstruct the possible horizontal gene transfer event or to trace the genome evolution. So in order to get something like this one, that I probably need to explain a little bit about how they do it. And this is the data matrix you have to have in the very beginning, and to have a particular group of taxa and to compare with another group of taxa and to compare the genomic similarity. And as you can find here, there's some group which is is similar, much more similar, and some of them are less similar. And this is a similarity test basically. And you can use a different cutoff value to show what is the core of the similarity taxa. So in this case, if you use a higher cutoff value for amino acid identity, you can identify a certain group which are highly similar to each other. For example, here to here. So that's the first thing you can identify. You can identify those, and then you can, based on that, to reconstruct another network. Basically, after you do that, then you have to identify the different individual genes and to compare those genes with each other, and then try to compare a very large data matrix together. And at the end, you'll probably find 3 different categories for those similarities among those different genes. And for the genome data, the present genome data is basically the very end of this display. If you consider this 3D structure, it will be the very top of the surface, and these are the genome data we have every point. And this is the cone-shaped like thing is the phylogeny eventually for reconstruct according to the time to the present time. And there are based on the similarity, you can connect the present in a particular case in a particular genome to reconstruction of the ancestral genome. And in this case, there are 5,000 cases for such things. You can connect the present genome to some place here or some place among the different genome. That's one kind of case. And some other categories, you can only connect those dots among. It never happened in the present day, but only in the ancestral state they can connect to each other. That's called the internal edges. And there are another category is that you can only find the similarity, the sequences to another genome, but not the common ancestor. So the third categories are those lateral gene transfer things. I mean, recent gene transfer things. So you can categorize into different network, and you put those things onto the top of the phylogeny. And also they develop a new algorithm. It's called a directed network. So for example, if we have a sequence here, there are 5 different sequences. How do we know the relationship among those sequences and the similarities, for example? So for example, right here, A has similarities to B and E, but not C and D. So you can make a data matrix like A to B and E with 1, and to C and D are 0. For the B, it's connected to all 4 of them, so except for them itself, it's 1, 1, 1, 1. So we have a new data matrix right here that's basically tell you the relationship or the similarity among those things. And you can imagine that, for example, it's there's some genes are shared by those 5 sequences, and you can have those genes to be connected to each other. But for certain genes, you can only connect those 3, but not the others. So that's one way to show it. So in a new algorithm, they want to know the directions for the gene transfer from here to here, or from here to here. Then you can use another algorithm to know the direction for the presence or absence of genes. And in this case, if you know it's from here to here or from here to here, then it's in here, outside here to B and D, you can press 1, but nothing is going inside A, so there's all 0 here. So you can create another network, and eventually you use this algorithm to calculate the relationship among those genomes, and you will probably get something like this one. And this is the outcome for those phylogenetic network. And this 1 dot represents 1 genome, and with a lot of connections from dot to dot. And these connections are based on the individual gene data. And for example, if we enlarge this part, it's on the right here, and you will see you can identify certain particular genes for these 5 genome, and from here to here you will have a gene transfer event, this CRIP-like transcriptional regulator from here to here, which means that those 3 has CRIP-like transcriptional regulator, and it's likely derived from here, but nowhere else. So you can use this to reconstruct the whole group of things. And those genomes share a lot of genes or share a lot of gene similarities will tend to group together. And then you can see there's some grouping among the different genomes. And there are a lot of different connecting points showing the directions for the gene transfer according to this one. So here, basically, this is just based on the genome similarity. Also on the content of what is the percentage of the genes they share, something like that, and they mark the bigger or the higher classification for the genome. And so this picture is showing that this grouping are highly correlated to our classification system right now. So which is a good thing, of course. This also means that the horizontal gene transfer cases likely happen. I mean, the lines are mostly within a certain group, but very few are connected the different color, for example. And this is a very powerful tool to identify the horizontal gene transfer in a very closely related cases. And if you consider a very long distantly related cases, it will be a little bit difficult to identify since there are too much problem. But if you are only consider a closely related group like this one, then it's very nice to visualize this event